have Carol Upchurch from Louisiana State University, and who's going to be talking about networks of PV neurons. And yeah, we're thrilled to have her here. Take it away.
Hi, everyone. My name is Carol. I'm in Dr. Canavier's lab. I got a lot of help on this project from Dr. Knowlton. And today I want to talk to you about persistent silencing of paravalbumin positive inhibitory interneurons and what that has to do with the subcritical Hopf bifurcation. To start off, though, why is this important? And it's something we think might be really valuable for working memory. So what I have on the left is an experiment done back in the 1980s. This is recording from the hippocampus. And what this experiment is they'd have a monkey pull down a lever, and then the monkey would get a cue. One or two buttons would light up. Then they needed to remember that cue for that period of time that we're seeing all this increased activity until later when they got a different cue that told them, okay, press the button and you get a treat if you remember which button to press. And what they identified from these recordings was a bunch of what they just called delay cells. And these delay cells were able to store the information from the cue in terms of right or left for that period of time, which could be, a couple of seconds, which is kind of long for neurons. And they just needed to store that information. It was part of this of storing things, working memory in this around this time scale. And this is from the hippocampus. I'm going to be comparing two models today, one from the CA1 region of the hippocampus and another from the interneuronal cortex. This is not behavioral data. This is straight ephys. This is people recording and injecting current. And you can see they would do a little depolarizing small amount of impulse, and some of these neurons would just keep firing for quite a while. So it was leading to this idea that for working memory, some neurons are just firing during that time where you need to keep something in your working memory to be able to use it. Some of y'all with really great working memory are wondering what my point is, because my slides were about persistent silencing. And that's what my next slide is. And this is a lot of really cool work from Dr. Chamberlain. And Dr. Chamberlain's, this paper presents one of the models that we're going to go over today. But what Dr. Chamberlain found was in the CA1 region of the hippocampus, a lot of paravalbumin positive fast spiking interneurons. If you gave them some current until they started spiking a lot, what you could do is interrupt them with a short hyperpolarizing little dip here. It was supposed to kind of be similar to like a synapse you would experience in vivo, but they'd give it this short little dip there and the neuron would stop firing for quite a while. But he took it one step further by looking at some of the pyramidal cells, some of the excitatory cells downstream from these paravalbumin positive fast spiking interneurons. And what he would do is he would inject just enough current into these pyramidal cells for them to fire around approximately about 2 hertz. And then he would repeat his experiments with the fast spiking interneurons with injecting current until they start spiking a lot and then interrupting them and having that really long pause that they just do. It extends much further than the interruption to see how that would affect the rate of firing in the pyramidal cells. And what you can see is if you inhibit these inhibitory interneurons, you disinhibit the pyramidal cells. So the rate of pyramidal cell firing during that interruption is higher. So the idea was that one way to be able to get to delay cells without, if it wasn't an intrinsic property of the delay cells themselves, was to cause an increase in firing by just decreasing the inhibition onto these delay cells, these cells that are firing more during that period that you need to hold something in your memory. So Dr. Chamberlain had a wonderful model in that paper to explain the behavior of his paravalbumin positive fast spiking basket cells. It was a Hodgkin-Huxley type, conductance-based model. Where's my pointer? With 4 different channels. The fast spike, your sodium channel that you need for spiking, KV1, KV3, and a leak channel. And KV1 ended up being really important for the behavior for the way that his cells fire. And what ended up being super important had to do with the kinetics of the inactivation gate, which it builds up at hyperpolarized potentials during like the after hyperpolarization, after an action potential, it builds up quite quickly at those hyperpolarized potentials where the time constant is relatively small. But it takes quite a while to relax back down at more depolarized potential because the time constant is so long. And again, this is a description of KV1. And to make sure that it was definitely KV1, he did a couple of experiments confirming that as well. So this is just staining for both parvalbumin positive, because it was parvalbumin positive fast spiking basket cells he was interested in, and KV1. So just making sure that, the cells have it. And, co-staining, and you can see the cells he was interested in and the cells that have this channel are the same cell. But he also did, some compelling electrophysiology work in terms of just blocking the channel, and the behavior goes away. You don't have such persistent interruptions in firing. So now we're going to dive into his model and how his model works. So again, it really has to do with that slow inactivation gate in his KV1 channel. That channel, it can build up during these deeper after hyperpotentials when the neuron is firing. So it starts to build up quite a bit. But it takes quite a while in order to relax back down to its original value during an interruption in firing. And then, of course, once you start firing again, you get those hyperpolarized potentials and you can build that back up again. To look at that through bifurcation theory, we have our bifurcation graphs on the side because H slow is what I'm interested in. H slow is going to be my bifurcation parameter. I love bifurcation graphs because I can fit a lot of information on on one graph. I'm going to start with the trajectory because I think it makes the most sense, or it's most intuitive. So for the trajectory, I actually kind of started out here when it seemed like the neuron was hitting its steady state limit cycle, and that's what I've highlighted here in blue. So I'm going to start from when it seemed to be at its steady state limit cycle. The interruption I graphed in red because I'm that is the trajectory on this. That is the trajectory of the interruption on this bifurcation diagram, but because the bifurcation diagram, the parameter is H slow, it assumes the level of current that I was injecting here. So I graphed it in red because I'm technically lying because the bifurcation diagram is not correct during the during the interruption and where I've graphed in red, because what should be happening is that the bifurcation would be moving towards the left, and that's what's causing the trajectory to fall onto this this set of stable fixed points. So now we can talk about the bifurcation itself. And what's going on is we have on the left we have unstable fixed points. So the fixed points are where your null lines of all your different state variables should be intersecting, but those can be unstable or stable that based off of the real part of the eigenvalues of a determinant there. But they're unstable over here until you reach a critical value of H slow where you'll have a Hopf bifurcation, and then they're going to be stable. Now you can tell that the trajectory, because my steady state limit cycle is kind of past that Hopf bifurcation. One of the things that I think are really cool about Hopf bifurcations is that even though you are changing the fundamental excitability of your system, you're not necessarily changing its behavior. And this is a great example of that, because H slow is still the neuron is still firing, H slow still builds up, and it gets quite past this Hopf bifurcation until it gets this steady state. And that's because with this Hopf bifurcation, you ended up with the formation of both stable and unstable limit cycles. So over on this side, we have in the dark black lines, I have the maximum and minimum voltages of the stable limit cycle. So where's my pointer? The maximum, being up here, and the minimum being down here. That's my stable limit cycle. And my stable limit cycle still exists past the Hopf bifurcation, so my neuron can still keep firing. What matters in terms of the neuron stopping firing is getting it to fall into this unstable limit cycle that was formed at the creation of the Hopf. If you get, again, this is we're taking a 5-dimensional system and projecting it down on 2. So just crossing it on this side doesn't mean you crossed into the space because of the way that it's projected. But your interruption causes you to cross into that space. And that interruption pulls you within the unstable limit cycle. And then you're going to get sucked down to this, where's my pointer? You're going to get sucked down to this branch of stable fixed points. And again, because it takes quite a while for H slow to relax back down to its original value, you're just going to spend time following that trajectory of stable fixed points while H slow slowly, inactivates. And then once you hit the Hopf bifurcation, again, Hopf bifurcation you're having where you're the real part of your eigenvalues goes from negative to positive. But I mean, it goes from negative to positive. It doesn't go from negative to like super positive. So it takes a little bit of time for it to kind of spiral out. And so it's hard to see on this scale, but it starts slowly spiraling out and then you start spiking again. Once you start spiking, you get those deep after hyperpolarizations. And again, this channel builds up during those after hyperpolarizations. That's what this purple line is for. It's the H slow null line. When you're under H slow null line, when you're below the H slow null line, you're going to be going towards it, which means you're going to the right. So during the after hyperpolarizations, your H slow is increasing and it increases a lot faster when you're hyperpolarized. And then when you're above the H slow null line, you're going to go back towards it. So you're moving towards the left, but you're doing it much more slowly, which is how the neuron, when it's firing, is able to actively build up H slow more and more until you again hit the point where the amount that you build up H slow during the after hyperpolarization is the same that you're kind of losing during the spike. The last thing on this bifurcation graph is the saddle node of periodics. This is when your stable and unstable limit cycle, collide and eliminate each other. That's not super important for this behavior, but it is super important for elliptical bursting. And I can get this model to elliptically burst in 2 ways. One way relates it to the paper that Dr. Chamberlain took. Dr. Chamberlain's model is a modification of a 2007 paper by Dr. Golem. So one way relates it to that older 2007 paper, and that is if you increase the conductance of KV1, you will move the bifurcation to the point where, where's my pointer? There it is. You'll move the bifurcation where the model will just kind of, at the right level of current, you'll cause H slow to build up until it hits that saddle node of periodics. And when those things eliminate each other, the only thing you have left is, again, that set of stable fixed points. You have to travel back down your stable fixed points, spiral out after you hit the Hopf, and then you'll start spiking again, but then you hit your saddle node of periodics, and that's how you end up getting this periodic bursting behavior. The other way to do that in his model is just to use a little bit less current so that the dynamics of H slow end up kind of dominating the system. But both of these, again, to get that elliptical bursting, you have to have it being pushed out onto the saddle node of periodics, which, like the interruption, means you're falling within that unstable limit cycle. Well, in this case, they're eliminating each other, but you get sucked into that trajectory of stable fixed points, and then you start to deactivate and then go back down to the Hopf and then spiral out. And that's how you can get repetitive bursting out of this model. Again, I wanted to relate this to the 2007 model that he took this model, adjusted some of the parameters for KV1 from, and that model explores the parameters of the window current of the sodium channel and the conductance of KV1 and of course their applied current. And you can get similar behavior if you increase KV1 enough. You can get this, they call it stuttering, but it's very similar to elliptical bursting. So that's kind of how these models relate to each other. This is a different model from Dr. Canavier's lab. This is from Dr. Villa, who was a postdoc in the lab a couple of years ago. I am skipping over a lot of really cool stuff about this model. Sorry, I'm skipping over a lot of cool stuff about this model. This model was done in conjunction with Dr. White, and it was actually a network model. We're only going to be analyzing one neuron, but it was Dr. White is an experimentalist, so they fit a bunch of different parameters to fit the spread of the data. But we're only going to be talking about one cell. And again, similar to Dr. Chamberlain's, it's a Hodgkin-Huxley type conductance-based model. Of course, we're not paying attention to the synapses right now because we're only considering its intrinsic behavior. But we've got a sodium channel, Kv7, Kv3, and a leak channel. We have Kv7 instead of Kv1, which means we don't have a slow and activating gate. We can still get, if you inject the right amount of current and then do a hyperpolarizing impulse in the same way, you can still get a persistent interruption and firing. But when we analyze this and trying to figure out what's causing this is me. What's causing this is me and my choices in the level of injected current that I've been using. So an important thing to notice about the bifurcation diagram is this is not a slope parameter that's intrinsic to the model. The slope parameter is the injected current that I'm choosing to use. And that's really important because while this model does go through a Hopf bifurcation and the amount of current I was injecting just had me just past the Hopf bifurcation, but at the point where I would be on a, where I could be on a stable limit cycle, the perturbation that I used pushes me out and then it ends exactly where I have it in. So I'd be right back. But again, when you're going through a Hopf bifurcation and the real part of your eigenvalues are going from negative to positive, you're going from negative to positive, but you're not going from like negative 0.0001 to 1000. So like it's positive, but it's small. So you end up having a really tight trajectory to spiral back out, which is what this inset is trying to what I'm trying to show with the inset. As soon as my interruption is over, I start spiraling out. It's just I'm spiraling out so tightly that it's taking a while to get out and back into firing. So these might both be from a Hopf bifurcation, but the first one is a parameter that's intrinsic to the model. It's the kinetics of that H slow, the inactivation gate of Kv1. This one, the Hopf bifurcation, is something that I'm forcing with the injected current. So that's going to cause some differences between the two. One, since there's nothing intrinsic and slow with the model itself, I shouldn't get elliptical bursting because if I'm injecting a steady state current, there's not going to be anything that's going to be pushing me backwards and forwards from the Hopf, and I don't have a subtle node periodics here to push out and then back through. And then the other way that these two models are different has to do with noise, and that gets a little confusing because the VIA model is a bigger, leakier cell. But if you have the noise scaled by the leak conductance of the neuron, Dr. Chamberlain's model is a lot more resistant. The persistent interruption is a lot more resistant to noise than Dr. Via's. And that makes sense because during this longer interruption, you have to travel across this trajectory of stable fixed points, and you have a little bit more room here in terms of something knocking you back out onto that stable limit cycle. It should be a little bit more resistant to noise than Via's model, which doesn't have that. There's no stable fixed points. As soon as you put the current back to something over here, you will be over here. I scaled the noise in two different ways. One of them was by the max current that I had injected, and the other one was by the leak conductance. The leak conductance makes a little bit of sense, but makes more sense in my opinion. But because Via's model was so much bigger, the leak conductance was a lot higher, so I scaled it by two different ways. But in conclusion, both of them go through a Hopf bifurcation, and that gives them a lot of cool characteristics, including bistability and having associated persistent long interruptions. But due to the fact that, like Dr. Chamberlain's, the Hopf bifurcation is an intrinsic parameter, it's intrinsic to the kinetics of a parameter of a state in the model. That interruption is going to be more stable to noise, and also does other cool behaviors like elliptical bursting. And again, why we think this is important is persistent interruptions and inhibitory interneuronal firing have implications for working memory by increasing the firing of locally connected pyramidal cells. So thank you so much for having me. I guess I'll take any questions.